Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Sam Moulton from the Business Review, and I will be your host. It's our pleasure to have VTT with us today, who will be discussing cell factories beyond fossil oil-based products. Today's guest speakers are Maria Pentila, Research Professor, Industrial Biotechnology, Laura Ruohonen, Senior Principal Scientist, Research Manager at Industrial Biotechnology, and Kirsi Maria Oxman-Kaudentie, Head of Research Area, and Juha-Pekka Pitkänen, Principal Scientist, Bioproduction Processes. I'd like to welcome you to our webinar platform on 24. You'll notice that it's browser-based. If you disconnect for any reason at some point, just click on the link you receive via email to rejoin. In order to ask questions, you can send these in via the Q&A widget at the bottom of your screen, or use the questions box at the top left-hand corner of your screen and type them in there. We'll try and allocate around 10 or 15 minutes at the end of the session to address any of your questions or thoughts. If you click on the red survey widget with white lines through, there are a few questions in there for you to answer in order to provide feedback, contact VTT, and give ideas for future webinars. You can do this during the webinar or immediately afterwards. If you click on the green resource widget, you'll find some links in there from our speakers to view. If you require help at any point, click on the yellow help widget. You can move all these windows around and resize and maximize them to get a better view of the slides. But now, without further ado, please let me welcome Maria.
Hello, and hello everybody there. It's very nice to be here talking to you in this webinar. So we are situated here in Espoo, which is very close to Helsinki in Finland. And for those who do not yet know VTT, so VTT is the leading research and technology company, government company. It's the leading one in Nordic countries. And from its beginning, VTT has providing, in particular industries, new technologies to increase their competitiveness. And this is what we are doing still today, and also networking globally with international companies and research organizations in doing this. Industrial biotechnology at VTT is very important, so we have a critical mass of experts, around 100 people in teams, metabolic engineering, synthetic biology, protein production, enzymology, bioprocess development, and plant biotechnology. So we are really covering a broad range of important biotechnologies. So our topic today is efficient cell factories, and we are going through the key steps in this presentation. The first is host organism selection, so that we get the best host for a particular product. Then very often the host needs to be still further engineered for the highest productivities and yields. And obviously we will test the organism in the production conditions, and then optimize the production to a larger scale, even towards the product, towards the production process. Our technologies are very topical today when we are moving towards bioeconomy, sort of biorefineries, where we use renewable plant raw materials, lignocellulosics, plant waste material, for making the products which are here on the right, which we today make from fossil oil. And that means that we need to convert the chemistry which we have in the raw material to the chemistries which we need for the product formation. And there are obviously very many different technologies needed. There is chemistry technologies, process technologies, and various engineering sciences needed in this. But one of the key technologies today is biotechnology, which is also considered very important at the European Union level. And when we talk about the chemical conversion from the raw material to the product, so cell factories, microbial production is very important. And this is because the cellular chemistry today can in principle already provide the chemicals which are today made from oil, but also very many chemicals for novel industrial applications through the specificity of cellular biochemistry. And it's good to note also that biotechnical processes can be very large scale, so over 1 million cubic meters. And this is, of course, something you need for bulk products like bioethanol, but also for butanol and lactic acid. And there are various different companies, chemical companies, and also energy companies who are turning to biotechnology for this reason. And there are some examples like DuPont, DSM, BASF, and the energy companies Total and BP. And obviously also biotechnology can be used for production of higher value products like pharmaceutical chemicals. And you have a table here which on the left shows some of the examples of commercial products from biotech today, and then on the right a few of the examples which are in the development towards an industrial process today. When we do our strain development work, so that needs to be, of course, compatible with the whole process. And if we start from renewable resources, so we need to be compatible with the pre-treatment, which is also carried out at VTT. The cellulose and hemicellulose are then digested to sugars, hydrolyzed to sugars, which are then substrate for the fermentation, where we use our production host which we have developed. And then obviously further down we have purification of the product. We can use chemical processes to convert that further and to polymerize that to biopolymers. So the two middle ones are the steps where biotechnology is the key thing. And in addition to the production host development, also a very important point is to develop and produce the enzymes which are needed for hydrolysis of the pre-treated material to sugars. And here we have a long history, and in the next slide, I have a few examples how we develop the enzymes to be suited for a certain raw material and certain pre-treatment method. So we search for optimal enzymes from microbe, metagenomic sequences, genomic databases. And then we look at their activities and select the best ones. But very often the enzymes may not be optimal as such, what you find in the nature, so we can also do enzyme engineering. And for instance, one important point today is the thermostability of the enzymes. And here we have demonstrated that you can even improve the thermostability of an enzyme which already is naturally quite thermostable. And then finally, we have a production host for the enzymes, which is Trichoderma, the filamentous fungus, which is probably the most famous industrial enzyme production organism today, and where we can then do expression of tailored enzyme mixtures as the customer wishes.
And let's now move on to the steps in the development of the production hosts. Overall, these steps can be divided into 1, organism selection, 2, organism development, 3, process development, and 4, scale-up and piloting, all steps where VTT offers expertise for its customers and partners. These 4 major steps of development we would now like to walk through with you in more detail. So let's first look into the way VTT has practiced the selection of the most suitable production hosts for chemicals. There are several aspects to consider when selecting the host for your desired product. If you are targeting for a so-called second-generation bioprocess, that is considering providing the raw material for the process among lignocellulosic biomasses or fractions or side streams thereof, toxicity of the hydrolysates produced from these resources may become an issue as to how well the chosen production host tolerates the hydrolysate. The product to be produced may in itself be toxic for the host. Well-known examples of such cases are acids, longer-chain alcohols, and lipophilic products, which may challenge the performance of the production host. The raw material range is also important to consider. If you target your bioprocess second generation, all the sugars present, that is both 6-carbon and 5-carbon sugars, need to be accepted to make the process feasible. Or you may want to consider alternative raw material sources, such as pectin, derived from, for example, sugar beet or citrus fruit, as the basis for your process. Furthermore, you may want to base your process on polymeric substrates, and organisms such as filamentous fungi, many of which are efficient producers of hydrolytic enzymes, may turn to be the optimal host in such cases. Finally, when setting criteria for selecting the host, as early as possible consideration of the final bioprocess targeted for large-scale industrial production is very advisable. For example, would a low pH process be favored to simplify and reduce the cost of the downstream processing and purification of the product? Or aeration is costly in large-scale bioprocesses, so could it be avoided? When sourcing for suitable host organisms, I would like to take the opportunity to introduce to you the VTT culture collection. It presently consists of around 6,500 microbial strains and plant cell lines, of which many are unique. Once you have set your criteria, screening of very likely a large number of possible candidates needs to be well planned and carefully executed to arrive into the best result. Initial screening of a large number of candidates is best carried out on agar plates. Inhibitors may represent the product itself, thus the screening assesses tolerance to it by the species tested. But it may well be another factor or component, such as, for example, growth on a particular carbon source. The plate screening targets for decreasing the number of the candidates, which are then tested for growth and substrate utilization, examples of important parameters to be considered when choosing the host. Finally, detailed characterization under more controlled cultivation conditions is carried out to preferably only 10 to 20 production host candidates. To finalize the host selection and end up in only a handful of so-called best candidates, the strains are ranked based on the various screening approaches carried out. When metabolic engineering is the target for further development, traits of the candidate, such as ploidy, genetic tools available, is the genome sequence, are genetic elements known, are expression vectors available, efficiency of transformation, what transformation markers are available, and how efficient is the homologous recombination of crucial importance if you need to delete genes from the genome to omit certain undesirable activities. Finally, even though carefully considering the potential for engineering the host, it is still advisable to pick more than one final candidate for the development, as unexpected hurdles may turn up at the early phases of the development process. To close up the discussion of selecting microbes as production hosts, I would like to bring up our experience with non-conventional yeast and filamentous fungi when developing hosts for production of, for example, organic acids. These species may offer advantages in terms of acid tolerance, capability to export the product, or other properties desirable for an efficient production pathway or production process to be developed. We have, during the course of our developments for production of various products, metabolically engineered well over 10 novel species for the first time.
We at VTT have also a long experience in utilizing unconventional eukaryotic host organisms, such as plant cells, to produce fine chemicals or ingredients for various industrial applications. For establishment of the plant cell culture line, the first and very crucial step is always the selection of suitable plant material and the right organ as a starting material for establishing a viable, well-proliferating cell culture. Through initiation steps, we obtain so-called callus culture, which is undifferentiated cell culture growing typically on solid medium. Then we can bring callus cells to suspension in order to grow cells faster and in large volume. VTT has also different plant cell lines, including unique Nordic dairy cell lines in our accredited cell culture collection, as mentioned before. One of our special expertise is utilization of cell cultures from wild Nordic berry species. Some of them are becoming endangered and many of them are not possible to be cultivated. Recent clinical studies have shown that Nordic berries contain many beneficial compounds, mainly phenolics and complex tannin derivatives, which are known to play an important role, for instance, in inflammation and oxidation, which are the key mechanisms in many chronic diseases. Cloudberry, which is shown on the left side of this slide, Cloudberry cells, for instance, have many biological properties, and they can be used as an effective cosmetic ingredient. And I will come back to that when we will present our case studies. Like any other host organism, also plant cells need growth and production optimization and process development. Cryopreservation to store the interesting cell lines needs normally to be developed separately for each individual cell line. Now with the science and new technologies, we are able to capture and enrich the sets of efficient compounds in cultivated cells and produce them in large scale for industrial applications. The possibilities are numerous, ranging from whole cells to isolated individual compounds, and the applications can be found in cosmetics, industry, pharma, or healthcare, food and feed, and also natural flavors and fragrances are today also of high interest.
Okay, we can say as a summary of the host selection that the host is very often critical for achieving high production yields, rates and titers, because it may have some intrinsic properties that are very useful. And in particular, the non-conventional organisms, such as non-conventional yeast or filamentous fungi or other microbes, may provide such beneficial features that are, at least at the moment, very difficult to engineer to the host, such as robustness, for instance, in production conditions, or sort of property to produce certain precursor towards our product. And there is, of course, lots still to be discovered in the natural biodiversity, and as we heard, so plant cells, for instance, are very unique opportunities to look for specialty chemicals. And one very important aspect is also what very many companies, when they come to ask is that they ask for a host organism which is not very much covered yet with IPR and patents. So having a new host organism gives the companies freedom to operate and for own IPR. So once we have selected the host, we then go for engineering of the host. And this is here the second step in our host development process, and that means that we need to reconstruct the metabolic pathways, and very often there we also use heterologous enzymes from other organisms to do that. And we will go through some examples what we have done for product formation, for engineering, for product formation, and there are different kinds of drivers for us here at VTT to do this. Of course, the primary driver is that customers come to us, industries come to us and say that, could you please produce for us this and that chemical? And then we go for developing the organism for the customer. Sometimes we see that there's a gap in the value chain. One example is, for instance, glycolic acid, which can be polymerized to PTA and which is a very good, having very good barrier properties. But glycolic acid, the precursor, is today made from fossil materials. And here then we can choose to develop a biotechnical process for glycolic acid production and then give the monomer to our chemists for polymerization. One of the things we have also been considering quite a lot is that what are the side streams available? And of course, xylose from biomass is a very abundant one, sugar, and also there will be processes coming, they are under development, where the different fractions from lignocellulosic biomass can be separated, like the C5, the xylose fraction being separated from the cellulose fraction. And this, of course, makes a demand to develop something unique, which you can obtain from xylose only. And many of the examples, or actually all the cases we are now presenting to you today, deal with production of organic acids for the various reasons we have mentioned of their importance. And then that means that the engineering of the microbes does not only deal with engineering for the product formation, but also you may need to engineer the organism for low pH tolerance, product toxicity, or for the inhibitors which you have in the raw material. But obviously the productivity of the product itself is always the primary goal. Our strain engineering efforts usually start actually in silico, so that we are looking at metabolic models. We look at the possible pathways towards the product, and also we go for looking through bioinformatics all the possible enzymes we could use for pathway engineering. Taking them from various different organisms, different cofactor specificities of the enzymes, and so on. And then when we go for pathway construction, we can of course today use very rapid technologies like Gibson assembly, but also the quite new technologies for genome editing like CRISPR. And here we have developed the technology, for instance, for industrial yeast, which is working very efficiently. And in the case, our production host actually happens to be a new one. So then, of course, we may need to start from scratch, building genetic engineering tools. And nowadays, of course, genome sequencing is cheap, so we can do that and also annotate the genome. And we have our own metabolic model reconstruction methods where you can in an automated fashion reconstruct the metabolic model of a new organism. And we are here then following the cycle, so that we design, we build the organism, we analyze how it works, and then of course very often it means that you have to redesign and go around and build again and analyze, and then can improve the productivity several fold through this process.
Let's look into the three cases Merja just described to you, and as to developing production hosts for organic acids. And the first case being that of lactic acid, the monocomponent of polylactic acid, which finds applications in biomaterials. VTT carried out a long-term development project with NatureWorks to develop a yeast production host for this particular acid. Yeast was identified as a desirable production host due to the fact that it allowed the development of a low pH process, which simplified the downstream processing. The joint development included screening for a suitable yeast host, engineering several novel yeast species and developing genetic tools for this purpose, comparison of lactate dehydrogenase enzymes to their performance in converting pyruvate to lactate, and engineering the yeast for xylose utilization. Titers of over 90 grams per liter of lactic acid were achieved during this development process. As our second case, we would like to present that of glycolic acid. As Maria mentioned, VTT has internally developed a value chain for this acid, meaning that we have developed its production by yeast fermentation all the way to polymer production. And the polymer having excellent properties, it finds applications as barrier material in packaging. Glycolic acid is a product derived from the so-called glyoxylate cycle, an intrinsic metabolic pathway interlinked with the TCA cycle of the yeast cell, as shown in the picture to the right. The various steps of the development have included comparison of different glycylate reductase enzymes responsible for the final conversion step from the glycylate to glycolic acid, an activity that needs to be introduced to the host. Furthermore, metabolic engineering has been carried out to disrupt competing enzymatic activities, shown in red in the figure, along the production pathway to enhance carbon flow to the final product. Additionally, different yeast species were assessed as suitable production host. Modeling of the cellular metabolism has, particularly in case of glycolic acid, been carried out in order to identify further targets for the engineering to enhance the production. Our third case takes a different angle, as Maria pointed out, and that is the viewpoint of raw material supply. We know that even though many plant materials, such as hardwood, are rich in xylon, the polymer of xylose, xylose is still inefficiently exploited in bioprocesses of today. However, as pretreatment and fractionation processes for plant matter are developed, xylose will be increasingly available, and we need to evaluate its use in bioprocesses. An example of the use of xylose is of course already available today, that of the second generation bioethanol plants, many of them up and running. But additional uses should be assessed as attractive options for the use of xylose. VTT has a long-term interest in developing bioprocesses, particularly now for the use of pentose sugars in various bioprocesses, as they are a source not competing with the food chain. In addition to bioethanol, we have developed production of lactic acid and xylitol from xylose. These developments have familiarized us with various xylose converting enzymes, such as xylose oxidizing dehydrogenases. When xylose is oxidized by xylose dehydrogenase, the monoacid xylonic acid is formed. It is similar to gluconic acid and suggested to have comparable properties and thus find applications similar to its 6-carbon counterpart. It could be used as a complexing agent and chelator in dispersal of a concrete, or it may find use in copolyamides, polyesters, or hydrogels. Furthermore, it has been demonstrated as a precursor for 1,2,4-butane triol and ethylene glycol production. VTT has developed efficient yeast production hosts for xylonic acid production, showing titers up to 170 grams per liter. The process is a low pH process, and the yeast shows tolerance to the product and inhibitor present in the raw material, hydrolysate. The development process has included engineering of several yeast species as the optimal host for the production. Additionally, several novel xylose dehydrogenases were identified and tested in the production host to rank their performance. improvement in performance in respect to the host strain, and the enzyme catalyst is shown in the figure to the right. Additionally, as mentioned already, the strains were engineered to tolerate an inhibitor present in the raw material, hydrolysate. We also carried out analysis at single yeast cell level and determined the transcriptome under production conditions in order to understand the physiology of the production yeast and to identify further rate-limiting steps in the production. The yeast Pichia cudria cevi was, during these studies, identified as the optimal host for xylonic acid production. The development of this yeast as a production platform has been very comprehensive and serves as a good example of an approach worth taking when choosing a novel non-conventional production host. It has included, in addition to the above discussed, for example, genome sequencing and annotation to ease the toolbox development for engineering. The production process has, in addition, been carefully studied alongside the host development to provide feedback for further host engineering. Prior to moving to discuss the development of a bioprocess, let me conclude. We hope that we have been able to convince you that industrial biotechnology is a viable and relevant option when developing production processes for chemicals based on renewable resources. The developments in technologies such as synthetic biology to engineer the metabolism and targeted production pathways in the selected production host have advanced very rapidly. This has allowed the expansion of the product range that can today be produced by cell factories, and importantly, they have fastened the engineering of the chosen host significantly. These facts make the whole development chain much shorter and the entry into large-scale production and to the market much faster. We need a comprehensive understanding of the metabolism to be able to efficiently engineer the selected host. Thus, modeling of the metabolism is of significant importance during the development. The biodiversity, both in terms of the microbial production host options and the catalytic enzymes of the chosen pathway, is far from being fully exploited in bioprocesses of today. Thus, data mining of the fastly growing biological data available is an important asset to be taken into account during the development. In all the three cases discussed with you today so far, targeting the development to a full value chain, that is, from a microbial production process all the way to the final product, has been emphasized as a strong driver, and we believe that this is a key factor in developing feasible bioprocesses for chemicals today and in the future.
Yes, then on our development path, we move to process development, where we test the developed organisms and optimize the productivity, titer, and yield in bioreactor conditions. And after the process development in lab scale, we scale up to pilot scale, let's say, for instance, in 1 cubic meter in our facilities. And this provides material for testing purification processes downstream and also data for evaluating the techno-economic feasibility of the process. So we proceed to process development, and here we focus on optimizing the performance of the earlier developed production organism in fermentations. So as inputs, we have the substrates that have been chosen to be used in the process as the raw material. So these substrates can be different carbon-6 or carbon-5 sugars, or mixtures of these in case of many hydrolysates. And certainly the substrate can also be something a bit more special, like galacturonic acid from pectin, the citrus peel, or methanol, or methane, or hopefully eventually even CO2. And in the fermentation itself, we seek the optimal conditions to maximize the output of the desired product, whether it's a fuel or bulk or fine chemicals. And in the fermentation, we find the optimal temperature and pH conditions and the oxygen level that the cells need during the process. And Certainly, like in the case of bioethanol, the anaerobic processes would often be the most favorable as being more economical. And then we also consider the solid content of the raw material that we have available and whether hydrolysis of the solid material is performed separately before the fermentation or simultaneously during the fermentation. And the cultivations provide transcriptomics, metabolomics, fluxomics, or omics data in general as a feedback to production host development, and performance data for modeling of unit operations and process concepts, and all for the techno-economic evaluations. And the main data we want to obtain from the cultivations are the specific and volumetric production rates, yields, and titers. So the specific productivity, so grams of product per cell per hour, is a target especially for the production host development in order to maximize the flux of matter through the cells. And the volumetric productivity, grams of product per liter per hour, so this defines how large production units are needed to fulfill the desired annual production. So this is a direct indication to capital expenses of the process. And yields of product from substrate have an impact on the operation expenses of production, and they may need some further organism development to minimize the side product. And titer, or the final concentration of the product, has impact on the cost of downstream processing. For instance, in ethanol production, it would become too expensive to distill the product if the concentration after fermentation is below 60 grams per liter. And in addition to culture conditions, also the product purification options need to be considered. In some cases, it is beneficial to remove the product during the cultivation, for instance by stripping it to gas phase, or by two-phase cultivations where the product escapes to oil phase from the water phase where the cells are. And next we have a look at the case examples from the viewpoint of process development. So in case 2 of the glycolic acid production, the challenge was the formation of side products, which meant that we needed to maintain the substrate level such that yield of glycolic acid was optimized. And in case 3 of converting xylose to xylonic acid, the challenge was that it was a low pH process with inhibitors from the hydrolysate, which had a combined effect on the tolerance of the organism. And in case 4 of producing cosmetic ingredients with plant cells, the challenge was the non-conventional production organism. And the process solution in case 2 of glycolic acid production was to use automated sampling and online analysis to enable feedback control for a fed-back process, which maintained the substrate levels at suitable concentrations and prevented accumulation of the side product. And here we obtained over 30 grams per liter concentrations. And the process solution in case 3 of xylonic acid was to provide host nutrient supply so that the cells had energy to maintain production at low pH with inhibitors present from a hydrolysate. And here we reached product titer of 170 grams per liter. And furthermore, in the xylonic acid case, transcriptomics data was provided from batch and continuous cultivations as a feedback to modeling and organism and process design.
Well, as I mentioned earlier, plant cells can be utilized as host organisms for chemical production or specialized low molecular weight secondary metabolites of plant origin or even heterologous proteins. VTT has experience in plant cell cultivation. We can obtain cultures practically from any plant, proliferate them in different forms, engineer them and grow them from lab scale to pilot scale. When working in the laboratory scale, one needs to optimize the growth conditions and the compound profile. If needed, the genetic engineering comes to the picture at this point. We have excellent facilities and know-how how to analyze and characterize our cultures. The next step in process development is moving from laboratory scale towards the pilot scale. This includes downstream processing too. It is very different depending on the industrial need. I will show you now our case study to produce cosmetic ingredients from Cloudberries for the industry. After we have obtained the optimal cell line, we move to bioreactor cultivation. Depending, of course, on the need of the biomass and the cell culture itself, the bioreactor is then chosen. Plant cells can be cultivated in steel tank reactors in diverse volumes. If the light is needed, the disposable bioreactors such as wave reactor can be used as shown in the lower circle on the left side. The production is followed by an appropriate downstream processing. In this particular case, our customer wanted to have lyophilized cell material as a raw ingredient, which then they formulated to suitable for the cosmetic skincare product. In this case study is based on our long-term collaboration with the largest Finnish cosmetic company called Lumene. The company had a need to utilize plant stem cells from their plant-derived Nordic wild nature. The VTT developed the technology, optimized the cell line, and processed and produced them the ingredients.
So here at VTT in Espoo, Finland, we have excellent bioprocess facilities with over 60 bioreactors ranging from 200 milliliters of small parallel bioreactors to 1,200 liters of our pilot bioreactor. And here in the upper picture, you can see our pilot bioreactors, and in the lower picture, our small parallel 1 liter bioreactors. And furthermore, we have corresponding downstream processing equipment for cell separation, cell lysis, and micro and ultrafiltration. And we have also sophisticated automated sampling and measurement instruments for monitoring the reactors in order to enable rapid online feedback for process development. We use mass spectrometer for off-gas analysis for most of our bioreactors, and with online HPLC we can monitor substrate consumption and product formation. For instance, glucose, ethanol, glycerol, and acetate can be measured in less than 10 minutes. And we can take samples automatically to cold bath, for instance, in nighttime, and then analyze them later offline in daytime. And we have also developed automated sampling and filtration from slurry high viscosity cultivations for online analysis and process control of these large reactors. And to summarize the key aspects of bioprocess development, so this provides environments where the cells can show their full optimized production potential after we have optimized the conditions. And in the glycolic acid case, we developed a feedback controlled fed batch which produced over 30 grams per liter of the product. And in xylonic acid case, we made a low pH process successful in producing over 150 grams per liter of the product. And in Cloudberry case, we met the customer need by enabling bulk production of shear sensitive plant cells. And overall, bioprocess development provides feedback to both strain selection and strain engineering. And furthermore, it provides input data to techno-economic assessment for the process of these options. And especially these productivities, yields, and titers are the key data that we provide from this step. And finally, we want to conclude that VTT is experienced in developing efficient cell factories. We can provide either the complete path or individual steps as has been presented. So we have very capable hands in performing the organism selection, so in harnessing the biodiversity and choosing organisms which have great tolerance for substrate and product toxicity. And of course, as mentioned, the IPR, or freedom to operate, is more and more important. And the next step, engineering of cellular metabolism to obtain desirable chemicals. So they're starting by computer models, calculating the optimal pathways and choosing the appropriate enzymes for the pathway. And then applying the tools for host engineering. And finally, development of the viable processes, providing feedback to metabolic design and data for evaluation of the process concepts. And we can, of course, combine these with the supporting activities such as process chemistry or life cycle analysis at VTT. So this concludes our presentation, and we thank you for your attention. And with these words, I hand over to Sam now for questions. Sam?
Thank you very much. So we've got a couple of poll questions to ask all the attendees first of all. It's very simple to vote on these. You just have to click on the circle next to the answer and press submit. So our first poll question I've got is, are you interested in developing a biotech process? And the answers are yes or no. So if you could just select an option now and then press submit to vote. I'll give you a few more seconds to do that. Just a reminder, we've got our Q&A coming up in the next few minutes, so if you could start thinking of questions and typing those into the box at the top left-hand corner as well, so we can answer those for you. Hopefully you've all had enough time to vote on this poll for the moment. I'll move us on to the results. And yeah, it's pretty substantial. 78.6% have said yes. So if you could just comment on this result before we move on to the next poll.
Yeah, it seems very, very promising, and obviously this is the way to go.
Okay, thank you. So our second question we've got is, do you see biotech as a competitive process option? Again, the answer is yes or no. It's exactly the same. To vote, just select the answer and then press submit. And just another reminder while you're voting here, we've also got a survey running in which you can leave feedback and give ideas for future webinars, contact VTT as well. So you can fill that out now, or after the webinar's finished, it will pop up full screen and you'll be able to answer that survey then as well. For the moment, I'll move us on to the results of this poll. And yeah, again, even higher, 92.3% said yes. So if you could just comment on this, and then I think you wanted to talk about opportunities to meet you, and then we'll move on to the Q&A section after that.
We are thrilled. You are our kind of audience. Thank you very much.
So here we would like to invite you to meet us here in Helsinki area in Finland. So we have the 11th carbohydrate bioengineering meeting coming up in a couple of weeks here in Espoo. And you can find more information at cbn11.org address. And then we have the 6th Nordic Wood Biorefinery Conference coming up on 20 to 22nd of October here in Helsinki, Finland. And for that, there's more information at vtt.fi/nwbc 2015. So you're welcome to join us in these two events.
Okay, thank you very much. We'll start off the Q&A section now. So yeah, like I said, you can still type in questions at the top left and press submit to send those in to us. We've got about 10 minutes to go, so I hope we get some in from you. The first question that we're going to go with now is, what makes VTT unique in the sector?
I think it was quite a heavy package which you obtained now of different technologies which we have. But I think one of the unique things is that we have really been working on industrial biotechnology for long and with industry. So we were actually working, doing biorefinery work before it became fashionable, or let's say before it became something we really need for the world. So we have been working from the 70s before our times, and so we have been working with yeast and filamentous fungi, and of course these are now the very important microbes which we have for biorefineries. And then all the time developing the new technologies, starting with gene technology already in very early 80s, 81 or something like that. So doing a lot of things, also including brewers' yeast work which is going on. And that we really have all the capabilities from gene to piloting in the same facility, and very good understanding what industry needs. So I think this is quite unique with the infrastructures we have, and also with then the other 600 people who are interested here at VTT, how to develop a sustainable future, sustainable technologies for industries, going from gasification and chemistry and so on. And of course, there we can start combining also the different technologies for integrated processes. We are also moving towards other organisms, have started activities like photosynthetic organisms, microbes, and also testing whether electrosynthesis could be a future technology. But these, of course, are still further away from real industrial processes.
Okay, thank you. You might have touched on this, but the next question we've got is VTT involved with synthetic biology?
Yes, one could say so. We have, for instance, which is directly applicable, is of course the CRISPR technology. And we have developed that, as I said previously, we have developed that also to polyploid organisms, so that we can knock out the allele, several alleles at the same time, and also several different genes at the same time. So this is, of course, one of the synthetic biology technologies. We are following that quite closely because we have been part of the industrial platform, for instance, of the Sundberg network, which is in the USA, which has been there for 9 years now, since the start of the sort of developments, as we call synthetic biology. So I think we are pretty well integrated also to the community, and we'll then take the developments which we see that would be useful really for developing also industrial organisms.
Thank you very much. Next question I've got is, what is the minimum value / volume of ingredient required to make this approach viable?
Okay, if this concerns to the plant cells which were presented, so it's very difficult to say because it depends very much on the application. So in the cosmetic side, the volume of ingredients is of course varying very much, and the minimum value, I would say also it's, let's say, low to medium. But if we move, for instance, to the pharmaceutical applications, the minimum value can be, of course, very much higher. So it's very case dependent, and that's why it's quite difficult to answer.
And from bulk chemical side, so bioethanol price or lactic acid price, it's at the moment it's something like 50 euro cents per kilogram, and that is, as we know, bioethanol economics is on the limit, and maybe they can get a little bit better price from lactic acid. But anyway, it's kind of the starting price is something around 1 euro per kilo which could be a viable price for bulk chemicals.
Okay, thank you very much. We've got a couple more questions. Next one is, does VTT claim any patent rights on work done on a contract basis?
So we, as I mentioned before, we have been working all the existence of VTT, which is now 75 years old. so we have been working with industries, so we understand quite well the requirements of industries in this respect. So it really is case by case, depending what type of work is done with industry. But very often let's say that in comparison, for instance, to the cases which you have, for instance, in United States and when people work with universities, I think we are much more understanding the situation that the industries are in their wishes to require IPR rights. But also, very often we then need to consider that we retain our freedom to develop the technologies, and of course we have some platform technologies which we need to be able to use and continue further. So it is a discussion together with the industry, and usually we end up with a sort of very good win-win situation with the companies.
Okay, thank you. I'll squeeze in a couple more, which is, what are the industrial sectors VTT is working with? You might have sort of covered this already, but that's the next question.
Yeah. It's, as a whole, VTT, of course, we are covering very many different industrial sectors, information technologies and manufacturing technologies. But with industrial biotechnology, because it's of course an enabling technology, so in principle it can be used in very many different sectors, and as we discussed, so there is also the energy industry is already interested in biotechnology, not only for fuels but also for chemicals. So we are really getting sort of ideas of a biorefinery here in the same way as we had oil-based biorefineries. So it goes for working with information technology companies, making modeling tools, then it's producing the chemicals for various different industrial sectors, materials, and so on. So I think it is really broad. We also work in the enzyme side. We can work with obviously enzyme producers, feed producers, textile companies, and so on. So it depends on the case, but very broadly, biotechnology is an enabling technology.
Thank you. And the final question we've got is VTT working with bacteria?
Yeah. Yes, we are working. Of course, E. coli is a host organism. Sometimes in metabolic pathway engineering, you look at the feasibility in bacteria first. And then now increasingly we are also looking at the properties of bacteria which you may not have in yeast, for instance, which are of course have been for long our favored organisms because they are so good in real large-scale industrial processes. But then there are organisms like which are interested for us if we think about using the one-carbon compounds like methane, for instance. So then you start at least the production processes with those organisms which already naturally utilize methane, and these are bacteria. And do you want to add?
Yes, so some gas fermentation project we mainly work with bacteria, and then also kind of electron transfers or microbial electrosynthesis. But these are, of course, kind of quite emerging technologies and quite a lot of basic research still involved there. And certainly we have also customer projects where we have, customer has the bacterial organisms, so in process development we work with bacteria.
And some can be quite exotic ones.
Okay, thank you very much. So that brings us to the end of the Q&A session and the end of the webinar. Just leaves me to thank Maria, Laura, Kirsi Maria, Juha-Pekka for what was a great presentation, and VTT for sponsoring the session. To the attendees, you'll receive an email shortly in about 2 hours telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We also had a question in, someone asking if they could get a copy of the slides. A PDF copy of the presentation slides will be in the resource list widget when you log in to the on-demand version, so you can find that there. Once the webinar ends in a couple of minutes, the survey that I mentioned will take its place full screen on your computer, and yeah, we'd appreciate if you took the time to answer the questions in there, which you can give feedback, give ideas for future webinars, and contact VTT and other stuff as well. We look forward to sharing further webinars with you, so please keep an eye on the website I just mentioned. Follow us on Twitter, which is @BRWebinars for daily updates, and join our LinkedIn group, Business Webinars. Apart from that, thank you once again, and I hope you all have a nice day.